giving introductions for seminars. But that one was really thoughtful. I myself am super excited for this opportunity to speak to you guys. I'm glad that Mark was able to pull me in, even though I had the time zone wrong. And I'm also super excited to get to interact with Mark, IRL a little bit, because I also think he is a wonderful voice on Twitter. I learned so much about tropical plant pathology and translational plant health from the educational resources that he tweets out. So I don't know, I think it's a wonderful community. So today I'm going to focus a bit on trying to understand the ecology of Ralstonia from their genetic diversity, their biogeography, and their host range. I'm going to move. There it is, the advance. I'm going to kind of skip the themes of the lab for time, but I do want to note that we have a lab YouTube, and I try to upload seminar recordings to it when I give Zoom seminars. So I have a few years old Zoom seminar on some really cool research we did doing 3D X-ray constructions of tomato plants during Ralstonia infection. So moving into Ralstonia, this is a bacterial plant pathogen, globally important, globally distributed. For its life cycle, it tends to infest soil, and it can survive for years to decades. Honestly, not much is really well studied about that, especially connecting that survival data to the genetic diversity of these bugs. I think that's a huge gap for people who have access to naturally infested fields, for us to really understand where Ralstonia is persisting. But they are able to persist in soil. They then sense the chemicals that roots exude as the plants grow. The bacteria will chemotax towards the roots, enter via wounds or natural openings, carve their way to the xylem, and end up colonizing the xylem systematically, which we have here. At some point, they colonize the plant to a sufficient level that the plants no longer can transport sufficient xylem sap. And sorry, just my cat opened the door and is now trying to say hello.
She wants to join, I think.
Yeah, so you might see some tails flying around. I'll try. I won't make any more comments about it. So the plants ultimately wilt in this infection, and throughout the pre-symptomatic and symptomatic phase, the bacteria will continue to shed from the roots of the plants, where they can go back out to the soil, encounter a new host, but also when there's precipitation or heavy irrigation, that water moving through the soil can move the pathogens with it. So they can contaminate surface water, and if those water sources get used in an untreated manner to irrigate fields, that can be a major way that these pathogens move around a region. Okay. And during the pandemic, I read a lot of the old classic literature around Ralstonia. And at that time, it was called Pseudomonas. And I really love this kind of rant that Ivan Budenhagen wrote in the conference proceedings for one of the international Ralstonia meetings. And so Ivan poses the conceptual problem. He says, bacterial wilt is two words, but one conceptual entity. Ralstonia solanacearum, likewise, is a single epithet. Although the literature is replete with biotypes and strains, it generally treats wilt as if there is one disease and one pathogen, created, I presume, by God, all at once, everywhere. We're still living in a creationist Linnaean descriptive recording of static phenomenon which occur around us in our approach to our disease research, no matter whether we are generalists, biochemists, or molecular geneticists. The reality, of course, is very different. There are many bacterial wilts, and there are many Ralstonia solanacearums. They've originated and evolved in wildly different places, and they have different capabilities with both native flora and introduced hosts, and presumably different capabilities with soils and environmental conditions. So I just love the energy in this off the cuff rant that I even published in the proceedings. But I also really love this final paragraph, thinking about pathogens are diverse, right? There's not one phenotype. And so it's important to keep in mind what is a model organism, and then what is the natural genetic and phenotypic biological variation.
Recording in progress.
And so I think I'm echoing a little bit.
Yes, that's
Maybe
a little bit of echoing. That's all right.
okay, so I'm just I'm gonna mute myself so I can't hear the echo, and I won't be able to hear. Okay, so in order to understand some of this genetic diversity of Ralstonia, it's important to keep this evolutionary lens in perspective. And so these pathogens have emerged from within the gram-negative bacteria of the Proteobacteria phyla, and within the family of the Burkholdeриaceae. Within the plant pathogenic wilt pathogens, there are 3 closely related species that form a species complex that we refer to as the RSSC, which stands for Ralstonia solanacearum species complex. And I like to think a lot about the ecology of these organisms. So the ecosystems where Ralstonia inhabits are mainly the soil, surface water, and the plant hosts that they are. And really, it's this plant host is like the only time these pathogens are probably proliferating. I think in the other ecosystems, they're holding on for dear life. And they're quite good at it, but they're not as good as they are at growing in plants. And so if we compare our plant pathogens to the evolutionary neighbors, we see that a lot of those are isolated from soil and surface water as well. There are some either extremely minor pathogens or some plant commensal strains that do colonize the rhizosphere of plants. But what's really interesting is that a couple of these lineages turn out to be really great at the ecosystem of potable water systems. So like they survive the chlorine treatment, they live really great in this like very low nutrient environment. And because those strains colonize the potable water system, they end up in very low abundance in most of our molecular biology regions. And they can end up, if people are doing sequence-based community profiles for looking at microbes in environments that especially the microbial biomass is low, if there's not much target to sequence, what actually ends up being sequenced are these very rare kit contaminants. And so Ralstonia is well known as being part of this kit microbiome, which is basically artifacts in people's data set. But because they colonize these potable water systems, there's also a few lineages that have unfortunately caused very severe infections in folks who are already quite immunocompromised. When these immunocompromised humans have gotten medications that have been filter sterilized, but the really small Ralstonia strains pass through those filters and end up being inoculated into a human host. And then a crazy fact is that there's 2 different lineages of these environmental Ralstonia that are permanent residents on the International Space Station. And they are actually colonizing the water recycling systems that exist on the space station. So, Ralstonia has gone to many places. But I'd say, compared to all those environments, our plant pathogens are still in a lot of places, but a little bit more down to earth, I guess. And so a big question in our lab, and I think the field broadly, is trying to understand the genomic and phenotypic diversity of Ralstonia. So we're gonna, my talk's gonna focus on a few really new things that we're doing in the lab to try to build out knowledge and especially build out collaborations. One of these focuses on trying to improve the genomic resources for Ralstonia. Honestly, I think the resources are not bad, but you can always have more genomic resources. So I want to keep improving them forever. We're working to try to get a better understanding of the biogeography of Ralstonia, and then also trying to carve that, some knowledge into that central question in plant pathology, which is understanding host range of plant pathogens. Okay, so in our lab, we train a lot of undergraduates and integrate them into our research and kind of have this joint research education mission. And I particularly like bringing new scientists in to do, fairly parallel, somewhat repetitive, more mundane work that helps us characterize this phenomenal genomic and phenotypic diversity of Ralstonia. And in the US, there's this like new-ish, like last decade or two, trend of trying to integrate research and education at the university level through these programs called CURES. And CURE is an acronym that stands for a course-based or cohort-based undergraduate research experience. And the elements of a CURE are that the student participants, student researchers, learn a variety of research skills. They uncover the unknown through real research. They do the scientific process of iteration, learning, applying, and repeating. And they also build their communication and collaboration skills. And so with this CURE model in mind, I realized we have a beautiful synergy between research needs and these undergrad teaching opportunities, and that's to build out resources for understanding the diversity of Ralstonia populations. So in our CURE, undergraduate students get 1 or 2 strains of Ralstonia. They extract the DNA. We submit it for whole genome sequencing. So far, we've been doing Illumina sequencing, but depending on how costs and the error rates seem to be going, we might be switching over to Nanopore soon. And then the students spend a lot of time playing with the data. To make it user-friendly, we use this platform called KBase, which is put together by the Department of Energy in the US. And the goal is to basically try to democratize some of these advanced bioinformatic methods so that biologists can use it without necessarily installing every single dependency in every pipeline. So these bioinformatic tools are installed on this cloud server that can be accessed with a free account on KBase. And so the students will immediately dive into exploring the data, assembling genomes, doing phylogenomics of their novel strain compared to other Ralstonia, and generally exploring gene function, genetic organization, things that relate to what they might be learning in their college classes. Last winter, we had a cohort of students work on a population that Sally Miller shared with us. These were strains that her former PhD student, Nagendra Subedi, had collected from Bangladesh mostly, and also a few strains from Nepal, which is his home country. And this beautiful data set, is one of those classic kind of waiting to be published units that I already am building up in my own lab. And so I reached out to Sally and asked if she had strains to share, and she suggested this population. And so it was perfect. We carried out whole genome sequencing of 24 isolates. And in pink here, I'm showing the new isolates from this collection compared to all the genomes of Ralstonia that are publicly available, with this just being phylotype 1. And the other phylotypes being compressed up here. I'm really new to population genomics and this kind of work, but looking at this structure, I see that there's a lot of clustering of the Bangladesh Nepal strains here in the tree, but there's still, some significant branch length here. So what this suggests to me is that this is probably representing some of the endemic population in Nepal and Bangladesh, and that those strains have been there for a very long time and have been diversifying, over the millennia, over the cropping systems. But then there's also, it looks like a few cases of some exotic strains of phylotype 1 coming in as new introductions after they diversified in some other location, which are represented by these groups. And then we also, prior to this, sequenced the genomes of a collection of strains from the 1960s that were collected by some of the first Ralstonia epidemiologists, Louise Kira, Ivan Budenhagen, and Ed French. And we targeted these strains because of we were trying to identify additional strains in this phylotype 2b4 group, which we're starting to build out as like a model for understanding host range in Ralstonia. And so this is a large part of the postdoctoral work of Dr. Natalie Aoun in our research group. And our goal is to publish these as genome announcements relatively recently. I think a lot of the raw data is already available on NCBI, but as soon as we can make it available is really the goal. And then we also, through working on this K-base platform, have made a public narrative, which is the K-base term for like a page. You can access it on from this URL. And maybe someone can type that into the chat so not everyone has to write it down. This requires a free K-base account. And then this particular narrative is probably stretching the memory limits of what the cloud server allows. So it does take a significant amount of time to load. Sorry, I might have a way to change that in the future, but right now it takes a very long time to load. And so there's a URL here, and I also have the URL in a bioarchive preprint. And I just want to highlight this really nice figure that Prul Sharma put together in a collaboration paper between Boris Vanitzer's group and myself. And it just really nicely demonstrates the taxonomic changes of Ralstonia over time. My personal take on taxonomy is that it should reflect evolutionary relationships. And so it's good for the modern names to reflect our current model of the evolutionary relationships of organisms. But it's useful to have records of what the old names were, so that if you're looking for classic papers, you know what to plop into Google to find the find the papers. And so this is a really nice figure for, especially that timeline, so it'll impact when the papers were published. I want to put a brief note out that there's a recent proposal to split up one of the species of the plant pathogens into 2 different species. It really should not be split. So I would recommend that people don't use this Ralstonia nicotianae taxon. We're in the process of compiling and submitting a rebuttal about this, where we've done a really robust genomic comparison of every Ralstonia strain that's available. And based on this, we think that the cutoffs in this proposal aren't really drawn at like the best place. And so we recommend keeping phylotype 1 and 3 in the same name, which is Ralstonia pseudosolanacearum, which is written here. I bring this up because NCBI was quite quick in adopting this newly proposed name. It's only been published since March. And so they renamed a bunch of genomes, and what is great is they're quite responsive. And when we showed them this evidence, they have started the process to undo it. But as of right now, like half the things are named Ralstonia nicotianae. Like it's kind of a mess. So probably don't need to know that unless you're confused. Okay, so moving on to the next portion to think about the biogeography of Ralstonia. I'm a super newcomer to understanding population genetics, anything, and Ralstonia diversity. But I was trained by Dr. Caitlin Allen, and she is quite an expert and collaborated with the person who probably knew Ralstonia the best of any scientist, which is Philippe Prieur, who spent most of his career in, I think, CIRAD in Reunion Island. And Philippe's group defined the phylotype and sequivar system in collaboration with Mark Fagan at University of Queensland. And so this system worked quite well, mapped to what was later designated the 3 species of the plant pathogens, with phylotypes representing major groups within those 3 species. And then, of course, evolution is fractal. There, you can go down diversity all the way down. The next level of diversity with the Ralstonia literature is sequivar, which stands for sequence variant. This is a pretty nice system for describing the genetic diversity of Ralstonia, and especially has been seminal work. And so this system has been widely adopted by global plant pathologists to characterize the population diversity of Ralstonia on different cropping systems and in different locations. But what I realized when I started my lab and I was trying to get a better sense of what is where, and trying to find the paper, realizing that this information is so valuable, but like many things in science, it's fragmented in the literature. So there's 300 plus papers on the populations of Ralstonia in different locations. And this is all, locked away into, this supplemental table, maybe a main body table, but there's not one central place to go to see all this information. And so, especially when the pandemic hit and we had to switch to remote work, we had an undergrad at the time, Kyle Chipman, who had to complete a research practicum, which was part of his degree requirement. And so I suggested to Kyle that he, read the literature and compile the information in a systematic way. He took this off to a beautiful direction. And when I saw the data, I was like, this is very cool. And also realized that, of course, people are continuing to publish. And so we're continuing to mentor additional students to consolidate data into this centralized database. And so what we record is as much information as we can get about the isolation of these strains. We record the taxonomic status, what host plant they were isolated from, and we try to break that down at multiple taxonomic levels. That's mostly been for me because I didn't know anything about plant taxonomy until this. So seeing those different levels lets me see how closely related the different hosts are. The location, if there's any information about the sequence to the isolate, whether it's a gene sequence or a genome accession, we record that, and at least one reference per strain. And so we have on the version that's available in bioRxiv right now, it includes almost 8,000 strains from 105 locations around the world. Oops, that's my alarm to make sure I don't miss this talk.
And you're an hour early.
And just now, students have added a bunch more strains, and we're in the process of curating that to release it. So from putting all this information together, we can start to see some big patterns. The first of which being 2 of the 4 phylotypes are way more likely to be reported in the population genetics literature. And that's phylotype 1 and phylotype 2. Phylotype 3 and 4 are much less likely to be reported. So this suggests the model, and I don't think I'm at all the first person to propose this, that phylotypes 1 and 2 are more widely distributed than phylotypes 3 and 4. That if they are more widely distributed and it's not just a sampling bias, there's explanations for how that could happen. One is the dumb luck approach of maybe because of patterns of how plants have been moved around through commercial trade. These 2 lineages have just gotten lucky. But the other one is that there could be intrinsic traits in those genotypes that make them more likely to be transmitted and more likely to be established once they're moved to a new location. And we don't know. This graph is taking, I guess I'll explain the dots on the next slide. The purpose I'm trying to show here is the model that my predecessors in the field described in terms of the origins of diversity for the different phylotypes. So the phylotype 2 is thought to have emerged and diversified within the Americas, phylotype 3 within Africa, and phylotype 1 within continental Asia, and phylotype 4 within Southeast Asia, especially the Indonesian archipelago. And what's really fascinating about being able to see and infer these origins of diversification is it really shows just how slowly Ralstonia moves to new locations, especially prior to human movement. This was not a pathogen that could hitch a ride on the wind or migratory birds. It's a pathogen that needed the wet soil to not die off, and so mostly they were diversifying in situ. But now if we look at the modern distribution of these isolates, apologize, my cat is banging on a wall. They love to do this when I'm on Zoom. So the modern distributions, a current undergrad in the lab, AJ Sandhar, wanted to develop his Python coding skills. And I was like, perfect, we have this big data set. Here's a couple buzzwords, go Google, see what you can do. And I'm like truly blown away by what he's come up with. So he found a package that can create these interactive HTML files that shows the distribution of the different isolates. And to look at it more clearly, it's easier to look at it one phylotype at a time. So here I'm showing the phylogenetic tree of Ralstonia. These are the plant pathogens. And I've highlighted phylotype 3. If we look at the location of where those are found, they are thought to have originated in Africa, and they seem to be still restricted to the Africa part of the globe. Overall, they're seem to be globally rare. And then this next group, the phylotype 4, seem to be mostly distributed in Southeast Asia, the Korean Peninsula, Japan, somewhere in continental Asia. I'm not sure if that dot's centered on somewhere or like actually represents a location, but it generally is in the Asian part of the globe. And then if we look at phylotype 1, so this is the lineage that's thought to have originated in continental Asia. There's currently a sporadic distribution of these strains on like most of the continents. And this probably has been through multiple, movements of lineages into new regions. We see a similar kind of pattern with phylotype 2. These originated in the Americas, and I'm being careful in the US to only circle the Southeast. The drier climates in this part tend to make it not very hospitable to Ralstonia. So it's this part of the Americas that Ralstonia is common in. And so they originated in the Americas, but do seem to be distributed to other locations, possibly more so than phylotype 1. And within this phylotype 2, there's a lineage of Ralstonia that has been historically designated as race 3 Biovar 2. This is what folks in Ralstonia are now trying to call the pandemic brown rock clade, because there's, if you want to hear a long rant, this term isn't very useful. And so, but clearly this is a clade that is pandemic. If we look at the genomic diversity, there's not much diversity, which is very, it's very consistent with epidemic genomic structure. And then also if we hover over the different locations and see which phylotype 1 strains have been present, AJ has it so that it has the strain name and the sequivar. And most of these strains outside of the Americas are designated as the 2B1-2 sequivars, suggesting that these, that this really is the pandemic lineage, which I think has been known for decades. So the sequivar system has a lot of strengths, but it is, like any other tool, subject to bias and downsides. And I was kind of curious about these. So I knew that this was a tool that was based on DNA sequence of a single genetic marker. It's a virulence gene that is conserved in Ralstonia. It's one gene. And what I didn't know much about population of bacteria, but the one thing I knew is that they do horizontal gene transfer, and that they can transfer and recombine, even housekeeping genes. And so a single gene phylogeny can be prone to some, errors if that gene has been transmitted horizontally amongst groups. And so in collaboration with Boris Venatzer's group, I was delighted for some brilliant bioinformaticians in his group, Parul Sharma and Marcella Johnson, to be able to help answer the question of, how well do sequivars match phylogeny if we build the phylogeny from a core genome sequence? Are sequivars predictive of where the strains will land on a phylogenetic tree built with, a larger data set? And to be honest, I kind of expected it to not work. There's a single gene marker and it's only 7 or 50 base pairs. But that's me being naive. This is a very validated tool. And what is exceptional is how well it works in phylotype 2. So here you can see each branch of the whole genome phylogeny corresponds to clusters of sequivars. And so this is really, nice. This is some nice robustness of the system. On the other hand, things are messier for phylotype 1. So, this sequivar was landing, at least in 2 groups, there was some uncertainty with the documentation, but many of these groups, judging by the arrows, are landing on slightly different, parts of the phylogenetic tree. And so overall, sequivars do seem to be kind of messy to look at the global phylotype 1 population diversity. I don't think they're without value, but they should be used with some caution. Comparing a sequivar 15 from one study to another study, it might not be as similar as you hope. I think that the low genetic diversity in phylotype 1, so if we look at this tree, this is phylotype 1 and it's quite flat. It's not perfectly flat, but it's, phylotype 3 here has a little bit more depth. 2 actually has all these different branches, and phylotype 4 has a little bit more diversification. But phylotype 1 has the lowest genetic diversity, which might be both a problem in terms of resolution of one gene, as well as it might increase the propensity for horizontal gene transfer to happen and become fixed in the populations. And then, of course, science is done by humans. I know I have made mistakes. And so there's always a chance for experimental error to be leading to some of the confusion in data sets. So I was just at the Ralstonia International Conference in Montevideo, Uruguay, a month or 2 ago. And I had the pleasure of meeting Gilles Cellier, who works at ANSYS, which is the French phytosanitation group. He trained with Philippe Prior, and he's probably the world expert on population diversity and epidemiology of Ralstonia. And what's exceptional is he advertised at that conference that he developed and published just a protocol for assigning sequivar to Ralstonia, because part of the problem with the human error is there wasn't necessarily a clearly written protocol for how to do this. And there's no funding for like a centralized unit to like check all the sequivar sites. So we all do our best, but some small mistakes can happen. And so his 2 main points are, of course, make sure your Sanger sequencing reads are high quality. And then there's also a really consistent way to treat those EGL sequences so that you make sure you're comparing apples to apples and building a robust single gene phylogeny. he also works a lot on additional methods for understanding population diversity of phylotype 1. So, I think if you want to work with this group, definitely, reach out to Gilles, but read his papers and get a sense of the strengths and weaknesses of the different tools. My really, novice armchair opinion, having not, been a leader in this field, is that some of the confusion as well could be that Philippe's group, including Gilles, tended to work a lot more with diverse populations of Ralstonia that were close to their home lab in Reunion Island. And so since phylotype 1 is kind of a group of invasive lineages in this region, the tools that they developed likely work exquisitely well for those populations. But they might need to be validated to work better for the populations that have been introduced into other locations, and definitely for the much wider genetic diversity that's present in the whole ancestral location of phylotype 1. But I don't know. But I think there's a lot of fun research for folks who are better at population genetics than me. So I can't, I still have the speakers muted, so I can't hear if you're talking, Mark.
It's okay.
Okay, so for the next part of my talk, I'm going to dive into the complexities of host range in Ralstonia. And I use the word complexity on purpose. This stuff is complex, and I think that's why everyone has been overwhelmed by it, including me. I want to start with a historical note. So there was an attempt to establish a classification system based on host range in Ralstonia. This system was proposed in 1962 by Ivan Budenhagen, and it was, a very impressive consolidation of the knowledge that he gained from, working around the world, especially with banana plantations, and getting a sense of how, the strains that he was seeing. That said, Ivan was not everywhere. And this system really doesn't work great, expanding to try to make it work for all the diverse Ralstonia that are present around the world. In that paper I highlighted with Perul as the lead author, we do have a paragraph where we kind of summarize some of the problems with the race system. And so I want to use this opportunity to share some of my opinions about the race system and host range phenotyping. And mostly it's good. I think that it is absolutely valuable and super useful for the plant pathology community to continue isolating strains and quantifying and comparing their virulence on different hosts. It is so important to publish that host range data, ideally both as summarized forms and also trying to future-proof it, trying to make it possible for scientists in the next decades to maybe do a huge meta-analysis to bring together more data than any one of us, can do in our lifetime. And then when possible, sequence and publish genomes so that there is genetic data that can be correlated to those host range phenotypes in the future. And I only have one note, which is after you do all this beautiful work, don't try to fit a square peg into a round hole. Don't try to look at the complexity of the host range data and try to assign a race. You probably will just end up with race 1 Ralstonia, which is the grab bag of, it's not a banana isolate and it's not a cold virulent isolate. So I'd say don't worry about the race typing. So something else that we're starting to get to understand in terms of host range for Ralstonia is looking at the literature database. So we can look at the database and get some estimate of what are the natural hosts where a given Ralstonia group has been documented to have been isolated from. This is still imperfect data. It's going to have sampling biases. But it's also, huge data that, like, keeping into account those biases, we might be able to see some patterns and at least develop hypotheses for future studies. And so if we group each sequivar of Ralstonia as one of these symbols, and just count the number of different species of plants that each sequivar has been isolated from, this is the distribution, at least from a few versions ago of the database. And what we see is that there's this one sequivar 4, that has the broadest known host range. And I'll talk about that in a second. But first, I want to highlight our sequivar 18. This is what I think of as kind of a typical Ralstonia group, where they tend to be found on a lot of our solanaceous plants, tomato, pepper, potato, and eggplant, and then maybe occasionally on some other groups. And so this is like a lot of the lineages of Ralstonia kind of have this host range. But if we look at this sequivar 4 group, which we can also call 2b4, these strains have been isolated from plant species in over 20 botanical families. And so, and we're showing some of them here. We've got cucurbits, the melons, the solanaceae like tomato, musa, banana, and plantain, some monocot ornamentals, whole bunch of weedy things and ornamentals found throughout this long tail. And so this group, oh, I guess I'll get to that. Sorry, I didn't really practice the last part of the talk. I never do that. But we're trying to get a sense of what are the genes that contribute to these beautiful phenotypes. What is allowing this strain to kill off this ginger plant, but makes it less good at killing off clove? Because there are limits to host range of the different groups. It's just complex patterns. So what are those genes? Our group is following in the footsteps of especially the model that Philippe Prior's group began to establish. He established the fact that there was this older lineage of banana-infecting MoCo isolates in sequivar 4. And Philippe identified a new epidemic lineage of sequivar 4 on melons in the French West Indies. And throughout his career, built up genomic and transcriptomic data sets to understand why these strains couldn't kill banana. They could all kill some solanaceous plants. These strains could kill melon, but these ones don't kill melon. And so what's nice about this model is that these strains are closely related enough to hope that we find some signal amongst all the genetic noise. So we might be able to eventually associate host range factors to explain these patterns. In my group, I was lucky to recruit an awesome master's student. So Jonathan Beutler actually was a small-scale turmeric farmer before he came to his master's, and unfortunately got some infected rhizomes of turmeric and just watched his crop completely melt in front of himself. So he packed up and decided to come back to school and was very motivated to study the energy of the enemy. And it was like a wonderful coincidence that I had just started a new Ralstonia lab at UC Davis, and he had just arrived as well. And so we weren't able to, solve turmeric or really improve management of Ralstonia in Hawaii. But we were able to get some better knowledge about host range of this 2b4 group. So for Jonathan's master's, we were able to collect 19 strains of this group from Caitlin Allen, as well as Dave Norman, who's at University of Florida. And then from the literature, we selected 3 hosts that all of these hosts strains in this group have been documented from. And we chose them because they're in different botanical families. These ones are, economically important hosts. And impatiens is kind of the wild card. We did that because we can buy impatient seed. And most ornamentals are clonally propagated, which is just harder for host range experiments, especially during a pandemic. And so Jonathan did a lot of plant phenotyping. for each of the host plants, he carried out 3 trials with 10 to 15 plants per trial. And he produced a lot of graphs that look like this, where we have each strain here. And then we show this disease development over time on tomato in red, banana in yellow, melon in green, and then impatiens in blue. And what we see, if we look at the patterns and compare it to the phylogeny, is that we have what's probably a pretty clonal group here that we're calling subclade B. And it was such a testament to Jonathan's precision, because you could basically put these plots right on top of each other, and all of those clade B strains were perfectly in line. And so, this genetic clonality also had phenotypic clonality. To be honest, not what we wanted on either side, but we didn't have the genomes before we started. So, it's a little bit less rich data than it could be. Ideally, for these kind of studies, you want a good amount of genetic diversity and a good amount of phenotypic diversity to do downstream correlations. No laser, sorry, the cat's meowing. And so this is the data that Jonathan produced for his master's. We've published this as a small publication. We're ourselves trying to mine it to see if we can find, genetic reasons, but we're also okay if anyone makes progress, because there's so many open questions in this field. And after Jonathan graduated, Dr. Natalie Aoun joined our group, and she's an expert in genome-wide association analyses. She got her PhD at INRA, where she worked on the plant side and characterized genetic variants of Arabidopsis, and how the different genes are associated with different levels of susceptibility to tolerance to Ralstonia. And in her postdoctoral work, she's coming over to the bacterial side and using parallel approaches to try to answer similarly big questions. So the basis of trying to get to understanding host range is that we're using some genome-wide association approaches. For these approaches, you need to have a phenotype that's quantifiable, ideally with some natural variation of that phenotype. You can't just have the same phenotype for everything. And then also some genomic variation. In this case, we're starting with gene presence absence in the pangenome of this Ralstonia population. While Natalie's been getting up to speed optimizing the statistical approaches, she's also adding to the data set by phenotyping these strains on a resistant cultivar of tomato from Philippe Prieur's group. We knew that there were resistance breaking strains within 2B4. And what Natalie has shown is that there's a range of these phenotypes across the 2B4 lineage. So that excites us to try to get a sense of how some of these strains are breaking this resistance. And I'm not a breeder, but I think this is one of the main sources of quantitative Ralstonia resistance that's been used in a lot of the breeding programs. And so now we're kind of at the, trying to convert this beautifully complex data into binary phenotypes and do gene the pangenomics and run the analysis platform to associate the genes with the phenotypes. Natalie did a trial of this. Our takeaway was that we weren't really satisfied with having done this analysis with draft genomes. So she just finished extracting high quality molecular weight DNA, and she's going to submit it for nanopore sequencing so that we can get complete genomes to repeat this analysis and have more confidence in the gene hits that come out. So with that, I'm going to move into the summary. So I briefly talked about the genomic resources we're building out for Ralstonia. I talked about the work that we're really building on the lovely contributions of our peers to try to consolidate knowledge on the biogeography of Ralstonia around the world. And laid some breadcrumbs of how we're trying to dive into the complexities of host range. And with that, I will turn on my speakers so I can hear questions. I hope I did okay on time. My timer isn't set up. And yeah.